Hi, everyone. My name is Nacho Sanguinetti, and I'm a postdoc at Harvard University working with Hopi Hoekstra and Nao Uchida. Today, we'll talk to you about behavior and different ways of making behavior exciting and thinking about behavior as an exciting thing. So, the title of my talk today is Behavior or What the Brain Does. You can follow me here in the details below. So, let's start. Why should we study behavior? I think like one of the easiest claims to make that I encountered like once upon a time on Twitter was Alan Camp, a person who has really inspired a lot of this work on like contributing to much better like open neuroscience teaching. He said because it might be useful to know what brains do if we want to know how brains work. And I really believe this. This is something that if we really want to understand how brains work, knowing what the output of the brain in terms of behavior is critically important and how this changes with different parameters, etc. So one thing that people that study behavior like me like encounter all the time is this idea, this pernicious idea of like, is behavior enough? Can you get like a good paper like studying just behavior? Is it, do you need to get into the brain? The other thing is like that people are super excited about all these techniques in like neuroscience, how you can record so many different neurons and to photon and neuropixels. And there is kind of like this idea that behavior is kind of like a bit technically boring and not as challenging as the other techniques. So I want to dispel some of those myths. So today I'm going to argue three things. First, that behavior is technically exciting and is really something that we should all be thinking about a lot more and how to make more exciting and how to make um, it more technically rich. Second, that behavioral analysis is computationally exciting, and I hope you have already had like a few examples of that throughout the course. And the third is that the brain itself can be a window to understand behavior. So let's start. How do we measure behavior? Generally, when you think about measuring behavior, you're thinking about imaging. So taking a representation of the movements of the animal in space. And this can be done in many different ways. This is a nice paper that reviews many different ways in which one can image behavior, be it by doing infrared recordings using video of the behavior of a fly, or using field cameras that allow us to, at the same time, record different angles of the same thing that we're viewing. Or there's thermal cameras that allow us to, for example, in this case, record from bats in the night that because they have this heat signature, we can be able to detect them from the background that doesn't have this heat signature and allows us to image them. And even in some cases, calculate their body temperature. There are other ways to image things, like, for example, using X-rays or using sonography. So there's many possibilities and many different ways. And how we measure this behavior in terms of how we image this behavior can be varied depending on the technique that we use, depending on the cameras that we use. This is a case, for example, in G of like a gigapixel camera that is really has a really high resolution that allows us to film behavior in very naturalistic settings, and then allows us to like zoom in into like very small regions of this camera of this of what is being filmed and analyze the behavior of specific animals in this environment in very detailed ways, as you can see here from the different blocks that corresponds to very small parts of this like overarching image. So there are many different ways to use imaging to study behavior. There's also attempts at like studying behavior in a much more detailed way using imaging. For example, you might want to image specifically different body parts of a mouse that is running across like uh, a tunnel, and you can image him from the bottom, you can image him from the side. You can even use something clever like a mirror to do both things at the same time. And this can allow you to measure like the different body parts and to see precisely and segment with computer vision the different body parts of the animal. As you will see in a bit, and as I'm sure you have seen in the course, some of those requirements have been met by like amazing computational models to study pose estimation without markers. But there's other ways of doing very detailed videography. For example, a lot of people have done work to put like cameras on like head stages of animals they're recording from, and these cameras can use an IR mirror to look at the eyes of the mouse and be able to eye track where the animal is looking at any given time. And this is a fantastic thing, and can also like we can also like track the whiskers and have a very detailed behavioral quantification just based on imaging. But this is like more of like what we traditionally know and what we traditionally think when we think about like recording behavior. But let's remind ourselves that behavior is much more than what meets the eye. So how else can we measure behavior? I'm going to give you here an example. This is an example of two fish, Adios. And I hope you are hearing the sound that is being made. If not, I'm going to play it again, showing sound. And this sound that you're hearing are the electric signals that these two fish are making. Two are electric fish. These are Gymnotus omarorum from Uruguay, from my native country. And this beautiful dance that they're making is not a mating ritual. It's actually an agonistic encounter. They're fighting for territory. So we can know a lot of their behavior and type of behavior that they're doing based on the electric signals, based on something that we can see with images. So to talk about electric signals, so what electric fish do is they generate an electric discharge that they can use to sense the environment around it and sense how objects change the currents that flow through their skin. But in communication, what they do is that they can use these signals to determine whether they're fighting or not fighting, and to determine who is winning in a certain fight. So there are two different types of signals. For example, sometimes fish, you can see here that there are two different electric organ discharges that you can see as these spikes. But then you can see this moment where there's the chirp, this like fast modulation of the electrical discharge. And this is a signal that the other animal can use to interpret like where they are in this fight. And another signal that is very common in this kind of like agonistic encounters, as you can see here, there's two fish that are like generating discharges. But then at some point in this region in the center, one fish stops discharging. This is called an interruption. And this is a signal for like a submission from one of the animals, telling the other animal that, okay, you win. You won this agonistic encounter. So the electric discharge is useful for navigating, but also for communication. And it's really an important part of behavior that we can measure in a completely different way in this case, but just putting electrodes on the water and measuring the electrical field that are generated by these amazing animals. So there's other modalities of behavior that we really should be taking a lot into account. In the case of people like me who study rodents, like we think of vocalizations as a very important output of the brain and a very important behavioral output. The animals are vocalizing, and this is important, relevant information about their world and how they're engaging socially. So in many cases, this is like generating sound with my vocal cords in some cases, or like with the vocal folds in rodents, or many other physical mechanisms, like in the case of the syrinx of birds. And sometimes like these behaviors can be like even in ranges that we cannot detect, what is called like the ultrasound frequency ranges that are not detected by the human ear. And for example, many examples of this, of where like sound behavior is extremely relevant, is pups. Pups generate this call, this isolation calls to track the mother to be brought back into the nest. There's also alert and alarm calls that different animals do to send communication signals about like potential predators in the area. There's also a lot of like vocalization during play and during like interaction. that is this appetitive positive vocalization that some rodents do. And there's also, of course, like the very famous work on bird songs that I'm sure all of you are very familiar with. Just to clarify, like in terms of rodents, in particular in terms of rats, rats have these very special vocalizations that are above 50 kilohertz that are called ultrasonic vocalizations that are very much related to this appetitive and pro-social behaviors in rats. And just to demonstrate it a bit, I'll show you here a video of a rat that is being tickled by an experimenter in the brain lab. This is courtesy of Shinpei Shiyama. And you can hear, and hopefully you can see in the spectrogram below, like how the rat is like emitting these vocalizations to being tickled, and really is following like the hand and really wants to come to the hand and be tickled. You'll see a couple of examples of this, of the rat chasing the hand. And you see like once the hand is gone, the rat is still vocalizing and is still very attentive. Here you can see that the rat actually follows the hand and really wants to like play with it as well. And during this following, the rat is also vocalizing and generating these vocalizations. So this is just an example of how like vocalizations are another dimension of behavior that is very important and very interesting to measure, especially when we're considering like social. And let's end that with that beautiful belly laughter from the rat. But behavior can be, as I say, multidimensional and can go into many different directions. One of it is that behavior can be very dirty, can be very scatological. Animals are leaving scents everywhere. They are urinating in the environment. They are leaving like pheromones in the environment. They're scent marking. Some animals have like special glands for marking the territories. And they also are really following all these smells and scents and pheromones, and they use them for a lot of different like important behaviors in the life, like for example for foraging, for hunting for different animals, for setting different territories or not going to the territory of another conspecific, and also for communicating certain things about ownership of like caches or ownership of different parts of territory. So behavior is very dirty, and there's a lot of like possible dimensions that we could study when we think about behavior. So this is just a very nice example of how somebody did try to study like behavior that is very dirty and very different and very goes through a completely different dimension, which is the behavior of urination. And what they did was to find a very clever and a very technical and technically different way of like recording this behavior. So what they did was to have these UV bulbs, so ultraviolet bulbs, and this bottom camera, and allowed them to, and a paper, and allowed them to film the fluorescent at the UV range, fluorescent urination of mice. And they even combined this with fibrophotometry or even like stimulation with optogenetics. So you'll see here a mouse that is receiving different frequencies of stimulation of an area that is controlling nutrition. And in the top, you'll see the behavior of the animal, what we would generally consider like the behavior. Like we could like use deep lab cut to track the animal. We could like follow the animal in space, see its pose, see how it's changing. But at the same time, on the bottom here, you will see the difference, the spots that are left once the stimulation is increased, and this we can see as as another dimension of behavior. In this case, the animal like urinating, and we can measure like the area in the paper that is urinated for different frequencies and in different circumstances. So just to show that behavior cannot be just like this very simple thing or like this very like straightforward thing that we think of where we're just like, oh, measuring like how an animal moves, but it can also like include like very technical and different ways of approaching and understanding a certain behavior. So this is another case that it goes completely orthogonal. We know that animals need to eat, need to consume food, and this is a beautiful device that is open hardware that you can buy. I think like you can also buy from OpenEthos, and this is a food dispenser that allows you to measure how much food is retrieved from the center of the arena. If you have like a mice that is interacting with this device, this fed device, you can see that the mice don't feed more or less than in the normal conditions. But you can study like a lot of things. Like you can study how much they're consuming during the dark phase versus during the light phase. And you can see like all the different, here are different mice and how many pellets are retrieved over days. And you can see how like the circadian rhythm of the animal for feeding moves over time. So feeding is also like an interesting behavior, and it's very orthogonal to just like the locomotion aspect of the feeding. You want to know exactly, precisely how many nutrients the animal is getting over time. So this is another way of like measuring behavior that is also technically different. This is another example that also goes very in a completely different direction. Just setting up the pattern here of how like there's many different ways of studying behavior. In this work, the people from the Max Planck, they studied a group of baboons moving in space using GPS tracking. We're using collars. You can see the baboon having a collar here in his neck. And they would track like a whole pack of baboons. And the interesting thing was that they found like a lot of natural experiments in which the whole group was going in a certain direction, but then two baboons would like spread into two possible directions. And then the group had to decide like where to go, if they would follow one, would follow the other, or if they would compromise. So in this case, we're showing like a compromise, which means that the whole group moves in like a middle direction between what the two baboons choose to do. But sometimes the whole group decides to choose one of the two possible directions. And then the other baboon needs to come back, retreat, and join the group. So then they did study this, like they found a lot of natural experiments where this split in the group happened, and they studied how the group is deciding, and they did this just by following GPS in a really large scale way. So this is another dimension of behavior that like a very macro and collective way of looking at behavior. So the other concept that I want to talk about in terms of like looking at behavior and thinking about behavior in a more extended way and in a more technically diverse way is thinking about extended phenotypes. So extended phenotypes are manifestations of genes that occur outside of the organism that possesses those genes. So they are the result of like a certain phenotype, but they don't happen within the animal itself. They're kind of like a construction based on the phenotype of the organism. And for example, one such case that it has been studied a lot in the lab where I'm currently at Hopi Hoekstra's lab at Harvard, is the creation of burrows. So in the lab, we study like Peromyscus mice, which are called deer mice, that are a whole genus of like 50 different species that live across North America, and they have different behaviors in terms of making these burrows. Here you can see the extended phenotype is just the hole that the burrow that the animal makes underground. And this can be measured by like filling the holes with foam and then recovering digging the whole foam back and measuring how much length was covered by the animal, and also like how big the burrow is itself. And as you can see here in the right panel, there are different species that have different, very different behaviors in terms of this extended phenotype. Some animals don't burrow. Some of these species don't burrow. Some species do these small burrows. Some species do burrows with variable tunnels, and some species do even burrows with a big nest chamber and an escape tunnel. So you can see the architecture of the extended phenotype of the animal gives us a lot of information of what the animal is doing in nature, and is really important part of the behavior. And I'll just give you a couple of two examples that I'm sure you're familiar about. One is like spider webs, like different species of spiders live in different ways, and some live underground, and some create these amazing spider webs. And the whole construction and the whole architecture of the spider web is actually an extended phenotype that one can study. In fact, I believe there's people like using different drugs to give to spiders and seeing how the drugs affect their extended phenotype. So the thing that you're measuring here might not necessarily be like the movement of the spider, but you're measuring the end result, the production of the extended phenotype of the spider web. Or in the case here to the right of these beautiful birds from my country and South America called horneros. They are called horneros because they build these little nests that are like look like ovens, like clay ovens, because they do this with mud and with twigs. And there's this very interesting recent study that has shown like that some birds have a left asymmetry for constructing this extended phenotype, while some other birds have a right asymmetry for constructing this phenotype. So these are just like a couple of things that I hope inspire you into thinking of the behavior of an animal as not only like the movements that it does, but like some of the results of what they do in nature. So how is analysis of behavior, now that we talked about how recording it can be exciting and different and variable, how is analysis exciting? So I hope throughout the course you have already seen a lot of this in terms of like the tools that we currently have available for analyzing behavior in a way that does not restrict the behavior of the animals, that doesn't use markers, that uses like deep neural networks in order to track different behaviors, like for example here the pulling of a lever on top, or this fly here. And there's different algorithms that do different parts of this scale of the behavior, but this has been like extensively covered. But as I said, like behavior is more than what meets the eye. So there has also been like an explosion of different algorithms that deal with other levels of behavior, for example, sound. So in 2019, Coffey et al. Published this paper about deep squeak. This is a method using convolutional neural networks to detect vocalizations of mice. So this allows you to not only detect, but classify different kinds of vocalizations into different vocalization type and separate vocalizations from noise. And has been a fantastic tool for people studying vocalizations that before were like just annotating vocalizations by hand and just going one by one and seeing what kind of type of vocalization we have. There's other newer methods, for example, that are implemented not in MATLAB, like this Deep Squeak was at the beginning. I don't know how it's working now, but there's Deep Audio Segmenter that is implemented in Python. This came out like last year, and it uses a little bit of a different kind of like neural network using temporal convolutional neural networks that allow us to deal with like how things change over time in an interesting way. So yeah, so just to show you that there are these other tools that you can like look at and try to implement if you're looking at a more dimensional behavior. So the final thing that I want to talk about is related to my own work, and is how the brain can be used as a source of information about meaningful behavior. So during my PhD at the lab of Michael Brecht here at the right, and in very tight collaboration with Annika Reinhold, a master student, and Konstantin Hartmann, a PhD student, what we did was to teach rats how to play hide and seek with humans. So we had this beautiful behavior where we had the rat that would jump into the start box and could go into one of two possibilities. In seek trials, the rat would be like closed into the start box, then the experimenter would go hide, would open the box automatically, and then the rat would need to come out, find the experimenter in order to get the reward. And the reward was not like food reward or water reward. It was just an interaction with the experimenter, just like I showed you before with the tickling example. And then the rat would be returned and it would start a new trial. And for hiding trials, the experimenter would leave the box open, stay next to the box, and kind of be counting and letting the rat go away, look for cover, hide, and then eventually find the rat, and then have this interaction. And we studied this back in 2019 and saw that rats are capable of learning this game in a very short time, and they're also capable of switching roles between hiding and seeking. So this is like an example of a rat playing the hider role. So you'll see the rat goes into the start box, Annika starts counting next to the start box, and at some point you'll see that the rat jumps out, so it knows that since Annika is close, then I need to hide. So then it goes and it hides in this box, and then Annika starts pretending like she's searching for the rat, just to make the hiding state a bit longer. And you'll even see that the rat now peeps out and tries to see like, where is Annika? And then Annika comes back, and then there's this like social interaction, just like it was mentioned and showed before, like with the ticketing case. And so yeah, so this is like a rat playing hide and seek, and we had this beautiful behavior, but we wanted to see like how much of what the brain was representing was related to the phases of that were part of the design of the game of hide and seek, but what other things the brain could tell us about what the area of the brain cared about in terms of the behavior of the animal. So then we used this approach in which we had the recordings of MPFC of prefrontal cortex activity, and we lowered the dimensionality of that activity into some continuous factors, and then we used a hidden markup model to identify different states of the cortex, different latent states that explain the activity of the neurons that we were recording. And then we saw how these states relate or not to the different behaviors during hide and seek, and to other possible behaviors that the animal could be doing that we as experimenters might have not been like really paying attention to. So this is how it actually works. So we have all these neurons that have this activity over time, and here you see a single trial of hide and seek, and you see how the activity between the neurons changes in the different phases of the game. And you can see the same thing with the different factors of like these low dimensional factors. And finally, here in the lower bar, you can see how the hidden markup model identifies very different states for the different phases of the game of hide and seek, as titled here at the top. So you can see that the phases change when the animal is going through different parts of hide and seek based solely on the neural activity. So let me remind you again, this method is only looking at neural activity. The fact that it coincides with the changes, transitions in the game, it means that there are state transitions in the prefrontal cortex, in the latent state of the cortex that are like happening at the moment of transitioning of the phases of the game. So this can be seen a lot more clearer here if we look at different at hide and seek in a full session, and we look at different trials. So here each row is a different trial of seek and a different trial of hide, and you can see how once we put all the trials that are actually of very different lengths, a trial can be like 20 seconds or it can be 200 seconds. Once we compress all these trials and time warp them to the same kind of like alignment of the phases of the game, we see that the PFC is actually the HMM is actually detecting states that are very consistent with the phases of the game, regardless of how long they're taking. So we see this very like state-like representation of the phases of the game during hide and seek. And we can see this across different sessions. These are 3 different sessions now that have this very similar state-like representation. But this was not all. We wanted to do more. Generally, in neuroscience, what we do is we record brain activity and then measure brain activity with respect to behavior. This is what is called, for example, a PSTH. So you have a certain event that is happening, and you do a peri-stimulus time histogram of what happened to the activity before and after the event. We wanted to do something similar, but using reverse physiology. So we wanted to see what would happen if we looked at behavior with respect to events of changes in neural states. So now the neural state is our event, and now we want to see what behaviors produced the neural state. So something like this. So we have the hidden markup model, and we have the different states as they happen in time during a trial. And then what we can do is for each of these states, whenever they happen during a whole session, do a collection of videos that tells us what the animal was doing when this state was active. And a good way of seeing that is, instead of like videos, which is like included in our paper and you can look at them, is to look at like a visual representation of all those videos at the same time. So what we did was to take what was different, what was changing from a background in each of those videos, and superimpose it in these video collections for the different states. So here you have, for 3 different states, what the animal was doing when those states occurred. So in the first state here, you can see in B, we can see like all these trajectories of the animal just running around the environment. So this was more of a running state, and this state was predominant like after the rat jumps out of the start box and also like during parts of interaction. Then there was this other different state that was very interesting that we were not expecting to see, which was a state that was predominantly active when the rat had a wall to its right side. This was a very specific state that we wouldn't have tried to study like what is going to be the neural representation of like a state of having the wall to the right. So we were not thinking about this, but the method that we used allowed us to go from the neural activity to the latent neural states and try to find what kind of behavior was making these neural states happen in the brain of the rat. Finally, this is the third one, where this state is predominantly active when the rat is inside one of the starting box, but also when the rat peeps out of the different boxes in the environment. This was something like completely remarkable. This is something that we were not thinking about studying. And I think like overall, this post that we can use the neural activity of a certain area to try to understand what that area is encoding for and what that area cares about with respect to the behavior of the animal. And perhaps we might see that in our collection of videos for a given state, we might not understand what's going on, but that opens a lot of questions of what other dimensions of behavior we should be measuring to understand what is making this area of the brain transition into different states. So with that said, my conclusions are that behavior is the output of the brain, and I hope you all agree with that behavior is technically challenging, and also it has a lot of room for creativity and for designing experiments in a way that allows you to test different behaviors for many different dimensions. Also, that behavioral analysis tools are revolutionizing our depth of behavioral understanding, and that we hope that more of these will come up in the future for many more dimensions of behavior. And finally, that behavior in itself is meaningful, and that what the brain is doing, what the brain is doing with respect to behavior can be found out from the brain itself. So we can use the activity of the brain to find out what is that area of the brain caring about in terms of the behavior of the animal. Or, for example, not only the behavior of the animal, of the statistics of the world. So with that, I thank you all, and I hope you're having a great course, and I say goodbye from here. Thank you.